Tonight's lecture is over Chapter 5, The Working Cell. So as we were talking about in Chapter 4, all cells have a cell membrane, and cell membranes are what we call a fluid mosaic. The gist of it is that cell membranes define the boundaries of a cell. They also determine the nature of its contact with the environment, and the cell is dynamic and constantly in a state of flux. And the major components of cell membranes are made up primarily of a bilayer of phospholipids that have embedded proteins, carbohydrates, glycolipids, glycoproteins, and cholesterol, at least in animal cells. Moving on, plasma membranes must allow certain substances to enter and leave the cell. It also must prevent some harmful materials from entering and some essential materials from leaving. In other words, plasma membranes are selectively permeable. In other words, they allow some substances to pass through, but not others. As I said on a previous slide, in Chapter 4, we studied the structure of the plasma membrane, and we described the major functions of the proteins embedded within the phospholipid bilayer. So in this section, we'll focus primarily on how the plasma membrane regulates the passage of materials into and out of the cell. We'll begin by discussing passive transport. So molecules are restless. The heat energy they contain makes them vibrate and wander randomly. One result of this motion is diffusion, the movement of molecules of any substance so that they spread out into the available space. Each molecule moves randomly, and yet the overall diffusion of a population of molecules is usually directional. In other words, beginning in an area of space where there is a dense population of molecules, they will diffuse directionally where they are less concentrated. For example, imagine many molecules of perfume inside a bottle. If you remove the bottle top, every molecule of perfume will move randomly about, but the net direction of molecules will be out of the bottle to fill the room. You could, with great effort, return the perfume to its bottle, but the molecules would never return spontaneously. Moving on to the next slide. For an example closer to a living cell, imagine a membrane separating pure water from a solution of a dye dissolved in the water. Assume that this membrane is permeable to the dye molecules, meaning that they can pass through the membrane. Although each dye molecule moves randomly, there will be a net migration across the membrane to the side that began as pure water. The spreading of a dye across the membrane will continue until both solutions have equal concentrations of the dye. When that point is reached, there will be a dynamic equilibrium with the many dye molecules moving per second across the membrane in one direction as the other. So diffusion across a membrane is an example of passive transport. Passive because the cell does not expend any energy for it all to happen. However, the cell membrane does play a regulatory role by being selectively permeable. For example, small molecules such as carbon dioxide and oxygen generally pass through more readily than larger molecules such as amino acids. But the membrane is relatively impermeable to even some very small substances, such as hydrogen ions and other inorganic ions, which are too hydrophilic to pass through the phospholipid bilayer. Passive transport is extremely important to all cells. In passive transport, a substance diffuses down its concentration gradient, a region which the substance's density changes. That is, the substance moves from where it is most concentrated to where it is less concentrated. In our lungs, for example, passive transport along the concentration gradients is the sole means by which oxygen, which is essential for metabolism, enters the blood, and carbon dioxide, a metabolic waste, passes out of it. Moving on, substances that do not cross membranes spontaneously can be transported via facilitated diffusion by specific transport proteins that act as selective corridors. Without the protein, the substance does not cross the membrane or diffuses across it too slowly to be useful to the cell. Facilitated diffusion is a type of passive transport because it does not require energy. As in all passive transport, the driving force is the concentration gradient. Moving on to osmosis. The diffusion of water across a selectively permeable membrane is called osmosis. Consider the case of a membrane separating two solutions with different concentrations of a solute, say the sugar sucrose. The membrane is permeable to water, but not to the solute. The solution with a higher concentration of solute is said to be hypertonic to the other solution. The solution with the lower solute concentration is said to be hypotonic to the other solution. Note that the hypotonic solution, by having a lower solute concentration, has the higher water concentration. Therefore, water will diffuse across the membrane along its concentration gradient from an area of higher water concentration, hypotonic solution, to one of lower concentration, hypertonic solution. This reduces the difference in solute concentrations and changes the volume of the two solutions. People can take advantage of osmosis to preserve foods. Salt is often applied to meats to cure them. The salt causes the water to move out of the food toward the region of greater solute or salt concentration. Food can also be preserved in honey because the high sugar concentration draws water out of the food. When the solute concentrations are the same on both sides of a membrane, water molecules will move at the same rate in both directions, so there will be no net change in the solute concentration. Moving on, solutions of equal solute concentration are said to be isotonic to each other. You may have seen this in terms on bottles of contact lens saline solution, which is formulated to have the same solute concentration as the fluid at the surface of the human eye, making it non-irritating. The survival of a cell depends on its ability to balance water uptake and water loss. When an animal cell, such as a red blood cell, is immersed in an isotonic solution, the cell's volume remains constant because the cell gains water at the same rate that it loses water. You can see that in the figure down below on the left-hand side. In this case, the cell is isotonic to its surroundings because the two solutions have the same total concentration of solutes. Many marine animals, such as sea stars and crabs, are isotonic to seawater, but what happens if an animal cell is in contact with a hypotonic solution which has a lower solute concentration than the cell? Due to osmosis, the cell would gain water, swell, and possibly burst, like an overfilled water balloon. A hypertonic environment is also harsh on animal cells. The cell shrivels and can die from water loss. For an animal to survive a hypotonic or hypertonic environment, the animal must have a way to balance an excessive uptake or excessive loss of water. The control of water balance is called osmoregulation. For example, a freshwater fish whose environment is hypotonic to its body has kidneys and gills that work constantly to prevent excessive buildup of water in the body. However, problems of water balance are somewhat different for plant cells because of their rigid cell walls. A plant cell immersed in an isotonic solution is flaccid, and a plant wilts in this situation. In contrast, a plant cell is turgid or firm and healthiest in a hypotonic environment with a net inflow of water. Although the elastic cell wall expands a bit, the back pressure it exerts prevents the cell from taking in too much water and bursting, as an animal cell would in this environment. Turgor is necessary for plants to retain their upright posture and the extended state of their leaves. However, in a hypertonic environment, a plant cell is no better off than an animal cell. As a plant cell loses water, it shrivels and its plasma membrane pulls away from the cell wall. This process is called plasmolysis, and this usually kills the cell. And you can see an example of turgor pressure at the bottom of the slide on the right-hand side. Moving on, In contrast to passive transport, active transport requires that a cell expend energy to move molecules across a membrane. In active transport, cellular energy is used to drive a transport protein that actively pumps a solute across a membrane against the solute's concentration gradient. That is, away from the side where it is less concentrated and toward the side where it is more concentrated. Membrane proteins usually use ATP as their energy source for active transport. Active transport enables cells to maintain internal concentrations of small solutes that differ from environmental concentrations. For example, compared with its surroundings, an animal nerve cell has a much higher concentration of potassium ions and a much lower concentration of sodium ions. The plasma membrane helps maintain these differences by pumping sodium out of the cell and potassium into the cell. This particular case of active transport, called the sodium-potassium pump, is vital in the propagation of nerve signals. So since we mentioned energy in the context of active transport, I'd like to take a few moments to define energy and talk about it. Energy is defined as the capacity to perform work. Work is performed whenever an object is moved against an opposing force. In other words, work moves things in ways they would not move if left alone. For example, imagine a diver climbing to the top of a platform and diving off. To get the diver to the top of the platform, work must be performed to overcome the opposing force of gravity. In the diver climbing up the steps to the diving platform, chemical energy from food is being converted into kinetic energy, the energy of motion. In this case, kinetic energy takes the form of muscle movement. What happens to the kinetic energy when the diver reaches the top of the platform? Has it disappeared? The answer is no. You may be familiar with the principle of conservation of matter, which states that matter cannot be created or destroyed, but can only be converted from one form to another. A similar principle, known as the conservation of energy, states that it is not possible to destroy or create energy. Like matter, energy can only be converted from one form to another. A power plant, for example, does not make energy, it merely converts it from one form, such as energy stored in coal, to a more convenient form, such as electricity. That's what happens in the diver's climb up the steps. The kinetic energy of muscle movement is now stored in the form of potential energy. Potential energy is the energy that an object has because of its location or structure, such as the energy contained by water behind a dam or by a compressed spring. In our example, the diver at the top of the platform has potential energy because of his elevated location. The act of diving off of the platform into the water converts the potential energy back into kinetic energy. Life depends on the conversion of energy from one form to another. If energy cannot be destroyed, where is it gone when the diver hits the water? It has been converted to heat, a type of kinetic energy contained in the random motion of atoms and molecules. The friction between the body and its surroundings generated heat in the air and then in the water. All energy conversions generate some heat. Although heat production does not destroy energy, it does make it less useful. Heat, of all energy forms, is the most difficult to quote-unquote tame, the most difficult to harness for useful work. Heat is energy in its most chaotic form, the energy of aimless molecular movement. Entropy is a measure of the amount of disorder or randomness in a system. Every time energy is converted from one form to another, entropy increases. The energy conversions during the climb up the ladder and the dive from the platform increased entropy because all the diver's potential energy was lost to the surroundings as heat. To climb up the steps again for another dive, the diver must use additional stored food energy. Well, how can molecules derived from the food we eat provide energy for our working cells? The molecules of food, gasoline, and other fuels have a special form of potential energy called chemical energy, which arises from the arrangement of atoms. Carbohydrates, fats, and gasoline have structures that make them especially rich in chemical energy. Living cells and automobile engines use the same basic process to make the chemical energy stored in their fuels available for work. In both cases, this process breaks the organic fuel into smaller waste molecules that have much less chemical energy than the fuel molecules did, thereby releasing energy that can be used to perform work. For example, the engine of an automobile mixes oxygen with gasoline in an explosive chemical reaction that breaks down the fuel molecules and pushes the pistons that eventually move the wheels. The waste products emitted from the car's exhaust pipe are mostly carbon dioxide and water. About 25% of the energy that an automobile engine extracts from its fuel is converted to kinetic energy of the car's movement. Most of the rest is converted to heat, so much that the engine would melt if the car's radiator and fan did not disperse excess heat into the atmosphere. Your cells also use oxygen to help harvest chemical energy. As in a car engine, the quote-unquote exhaust is mostly carbon dioxide and water. The quote-unquote combustion of fuel in the cells of your body is called cellular respiration, which is a more gradual and efficient burning of fuel compared with the explosive combustion of an automobile engine. Cellular respiration is the energy-releasing chemical breakdown of fuel molecules and the storage of that energy in a form the cell can use to perform work. We will discuss the details of cellular respiration in the upcoming chapters, but you convert about 40% of your food energy to useful work, such as the contraction of your muscles. About 60% of the energy released by the breakdown of fuel molecules generates body heat. Humans and many other animals can use this heat to keep the body at an almost constant temperature. Even when the surrounding air is much colder, the human body and the bodies of other animals stay at a constant temperature. The liberation of heat energy also explains why you feel hot after exercise. Sweating and other cooling mechanisms enable your body to lose the excess heat as a car's radiator keeps the engine from overheating. Read any packaged food label and you'll find the number of calories in each serving of food. Calories are units of energy. A calorie is the amount of energy that can raise the temperature of 1 gram of water by 1 degree Celsius. You could actually measure the caloric content of a peanut by burning it under a container of water to convert all of the stored chemical energy to heat and then measuring the temperature increase of the water. Calories are tiny units of energy, so using them to describe the fuel content of foods is not practical. Instead, it's conventional to use kilocalories, that is, units of 1,000 calories. In fact, the calories, which you see on a food package, capital C, are usually in kilocalories. That's a lot of energy. For example, 1 peanut has about 5 kilocalories. That's enough energy to increase the temperature of 1 kilogram of water by 5 degrees Celsius in our peanut burning experiment. And just a handful of peanuts contains enough calories, if converted to heat, to boil 1 kilogram of water. In living organisms, of course, food isn't used to boil water, but instead to fuel the activities of life. Calories are burned off in some typical activities such as exercising. The carbohydrates, fats, and other fuel molecules that we obtain from food do not drive the work in our cells directly. Instead, the chemical energy released by the breakdown of organic molecules during cellular respiration is used to generate molecules of ATP, adenosine triphosphate. These molecules of ATP then power cellular work. ATP acts like an energy shuttle, storing energy obtained from food and then releasing it as needed at a later time. ATP consists of an organic molecule called adenosine plus a tail of 3 phosphate groups. The triphosphate tail is quote-unquote the business end of ATP, the part that provides the energy for cellular work. Each phosphate group is negatively charged. Negative charges repel each other. The crowding of negative charges in the triphosphate tail contributes to the potential energy of ATP. It's analogous to the storing of energy by compressing a spring. If you release the spring, it will relax, and you can use that springiness to do some useful work. For ATP power, it is the release of the phosphate at the tip of the triphosphate tail that makes the energy available to working cells. What remains is ADP, adenosine diphosphate, 2 phosphate groups instead of 3. ATP is sometimes referred to as cellular currency. As you've seen, a living organism contains a vast collection of chemicals with countless chemical reactions constantly changing the organism's molecular makeup. In a sense, a living organism is a complex quote-unquote chemical square dance with the molecular quote-unquote dancers continually changing partners via chemical reactions. The total of all the chemical reactions in an organism is called metabolism. And I would define death as the loss of the ability to metabolize. Interestingly, almost no metabolism occurs without help. Most require the assistance of enzymes. Enzymes are proteins that speed up chemical reactions. All living cells contain thousands of different enzymes, each promoting a different chemical reaction. For a chemical reaction to begin, the chemical bonds in the reactant molecules must be broken. This process requires that molecules absorb energy from their surroundings. This energy is called activation energy because it activates the reactants and triggers the chemical reactions. Enzymes enable metabolism to occur by reducing the amount of activation energy required to break the bonds of reactant molecules. If you think of the requirement for activation energy as a barrier to a chemical reaction, an enzyme's function is to lower that barrier. It does so by binding to reactant molecules and putting them under physical or chemical stress, making it easier to break their bonds and start a reaction. So now that we've discussed energy a little bit, let's move back to active transport. Active transport requires energy and pumps. To move substances against a concentration or electrochemical gradient, the cell must use energy. Active transport mechanisms, collectively called pumps, work against electrochemical gradients, and there are 3 types of pumps. We see in the diagram or the cartoon below, we have a uniporter, which the solute moves one way in one direction. We have a symporter where 2 different molecules can move through the same protein but in the same direction, and we have the antiporter where 2 different molecules move in different directions through that protein. Moving on to primary active transport, where ions move across a membrane and create a difference in charge across that membrane. This is directly dependent upon ATP, and we can see that illustrated in the cartoon below. So the gist of this cartoon is that moving the ions against their concentration gradient creates an electrochemical gradient. Moving on to secondary active transport, this brings sodium ions and possibly other compounds into a cell. As sodium ion concentrations build outside of the plasma membrane because of the action of the primary active transport process, an electrochemical gradient is created. If a channel protein exists and is open, the sodium ions will be pulled through the membrane. This movement is used to transport other substances that can attach themselves to the transport protein through the membrane. Many amino acids as well as glucose enter the cell this way. This secondary process is also used to store high-energy hydrogen ions in the mitochondria of plant and animal cells for the production of ATP. The potential energy that accumulates in the stored hydrogen ions is translated into kinetic energy as the ions surge through the channel protein ATP synthase, and that energy is used to convert ADP into ATP. Moving on. So far, we focused on how water and small solutes enter and leave cells by moving through the plasma membrane. The story is different for macromolecules such as proteins, which are much too big to fit through the membrane itself. Their traffic into and out of the cell depends on the ability of the membrane to form sacs, thereby packaging larger molecules into vesicles. You have already seen an example in the packaging and secretion of proteins. During protein production by the cell, secretory proteins exit the cell from transport vesicles that fuse with the plasma membrane, spilling the contents outside of the cell. That process is called exocytosis. When you cry, for example, cells in your tear glands use exocytosis to export salty tears. The reverse process, endocytosis, takes material into the cell within the vesicles that bud inward from the plasma membrane. There are 3 types of endocytosis, and the first one we'll discuss is phagocytosis. In phagocytosis, or cellular eating, a cell engulfs a particle and packages it within a food vacuole. The next type we'll discuss is penocytosis. In penocytosis, or cellular drinking, the cell quote-unquote gulps droplets of fluid by forming vesicles. Both phagocytosis and penocytosis are nonspecific in the substances transported. In contrast, receptor-mediated endocytosis is very specific. It is triggered by the binding of certain external molecules to specific receptor proteins built into the plasma membrane. This binding causes the local region of the membrane to form a vesicle that transports the specific substance into the cell. For instance, human liver cells use receptor-mediated endocytosis to take up cholesterol particles from the blood. And this next slide outlines cellular transport. It gives you a nice table that shows the methods of transport, their energy requirements, and the types of material that are transported. So as you can see in the first column, we start with diffusion. It's a passive process, and it transports small molecular weight material. So I'll allow you to review this table on your own time. And we'll wrap up with a quick review of tonight's lecture. So since you've listened to the lecture tonight, you should be able to know why cell membranes are said to be fluid mosaics. You should also be able to list the major components of cell membranes and their roles. You should know what the types of passive transport are. You should be able to describe which direction water will move in solutions of different tonicity. You should be able to define 3 types of pumps used in active transport. You should also be able to define energy and ATP. You should know what types of active transport there are. You should be able to define 3 types of endocytosis. And you should know what the steps are for exocytosis.